Hello students, today I want to discuss uh, some questions from microbial cell culture and its application that may come in your CBSE exam. So what is difference between primary metabolite and secondary metabolites? Such microbial products that are produced during exponential growth phase. There are three phases of microbial growth: S phase, lag phase, S phase, and stationary phase. So sigmoid phase, that is known as exponential growth phase, during which there is a production of primary metabolites. But if you observe the production of secondary metabolites, it always occur during stationary phase. So these are the major difference between primary metabolite and secondary metabolites. Now try to recall examples. What are the primary metabolites? Ethanol, citric acid, vitamin C, vitamin B12. These are the primary metabolites. And secondary metabolites are toxin, antibiotics. Next, I want to discuss this. What is difference between isolation of intracellular microbial metabolites and extracellular microbial metabolites? So try to difference during isolation. If you observe the characteristic feature, after centrifugation, if you inoculate the microbial culture of the two different types, one is intracellular microbial metabolites, means that bacteria produce the metabolite inside the cell, and extracellular microbial metabolite means that particular bacteria produce the metabolites, means chemical compound outside the cell, means inside the media. So when you inoculate, after incubation, we do the separation of the bacterial cell and media. So after centrifugation and filtration, in case of isolation of intracellular microbial metabolites, we are concerned with microbial cell, not for the media. But in case of extracellular microbial metabolite production, we are concerned with filtrate of the media, not bacteria. That is the major difference. When you are crushing the microbial cell, in which case that is required? In case of intracellular microbial metabolite, it means whatever the useful compound, that is the microbial products, it is present inside the bacterial cell, so you have to crush it. That is required in case of isolation of intracellular microbial metabolites. That crushing of microbial cell is not required at all, because we are going to discard the bacterial cell. Discard, in case of discarding filtrate of media after isolation of bacterial cell, we are going to discard during production of in isolation of intracellular microbial metabolites. In case of extracellular microbial metabolites, we are going to discard the microbial cell after getting protein in the media. Means we are concerned 100% with the media. If you observe the process, centrifugation, it required two times. In case of isolation of intracellular microbial metabolites, in this case, for isolation of extracellular microbial product, that is one time required. Biochemical techniques, if you observe, then cell lysis, centrifugation, concentration, purification, such type of thing is required. But same process like filtration, liquid extraction, chromatography, that is required in case of extracellular microbial metabolites. We do the production of insulin from E. coli bacteria in case of isolation of intracellular microbial metabolites. But in case of extracellular metabolites, we produce the Penicillin. It is an antibiotic produced from Penicillium chrysogenum or Penicillium notatum. Now come to next question. What is doubling time? It is very important for your coming CBSE exam. And what is the relationship between doubling time and growth rate? And derive the equation for the doubling time. So first of all, try to know what is the doubling time. The minimum time in which bacterial population or mass of bacteria become double is known as doubling time. So there is inverse relationship between doubling time and growth rate. It means if there is a more doubling time, growth rate will be less. If there is a less doubling time, growth rate of bacteria will be more. So this relationship is valid for microbial culture, animal cell culture, plant cell culture. So try to know the equation. TD means doubling time equal to 0.693 by mu. So that is very important, where mu is a specific growth rate. You try to know the symbol of mu. Okay. So there is an inverse relationship between doubling time and a specific growth rate. You can differentiate the microbial culture and try to derive the equation. So many times that type question has been asked in your CBSE exams. Next topic is very important for your coming CBSE exam. Metagenomics. What is metagenomics? Metagenomics is the study of metagenomes, genetic materials recovered directly from environmental samples. We directly take the environmental samples and try to find out what are the useful genes present in that metagenomes. So it describes as comprehensive study of nucleotide sequence, the sequence of the gene, a structure, regulation and function of the genes. Scientists can study the smallest component of an environmental system by extracting the DNA from an organism in the system and inserting it into a model organism, which then express this DNA where it can be studied using a standard laboratory technique. So first of all, scientists try to isolate the useful gene from any unknown organism, then try to insert in any model organisms, and then they study what are the different possible function of particular genes. Next, that is metagenomics is employed as a means of systematically investigating, classifying and manipulating the entire materials isolated from environmental samples. Means we are going to study systematically and classify and manipulate the entire genetic materials. It may be useful for human beings. So this is a multi-step process that relies on the efficiency of four major steps. What are the four major steps? First is isolation. First is isolation of genetic materials. Second is manipulation of genetic materials with the help of restriction enzyme, DNA ligase, and then construction of DNA library. That is very important for getting many copy of particular gene of interest, and then analysis of genetic material in metagenomic library. So this metagenomics represent a powerful tools to assess the bonding diversity of native environmental samples. Means we are taking the sample directly from environments and try to find out whether the useful gene are present or not. Through the many molecular technique, you can isolate the gene, sequence the gene, and then try to find whether that gene is useful. Then we can clone the gene. So the valuable property of metagenomics is that it provides the capacity to effectively characterize the genetic diversity present in the samples, regardless of availability of laboratory culture techniques. So just try to know what is lyophilization. What do you mean by lyophilization? It is a method of preservation in which water is removed by sublimation from any microbes. What do you mean sublimation? Conversion of liquid water directly into vapor form. Okay. In presence of cryoprotective agent, sealing in a ampoule that can lead to long-term viability with 30 years have been reported. Means we can store any microbial culture up to a 30 year, but after lyophilization, it is a method of preservation in which water is removed by sublimation in presence of cryoprotective agent. So try to know what is cryoprotective agent. That prevent the formation of ice crystal inside the bacterial cells. Okay, so that's why that sample will remain viable for a long period of time in a dormant stage. In lyophilization, cells are stored as lyophilized cell under vacuum condition at very low temperature, either 5 degree or minus 20 degree up to 70 degree. So lyophilization remove water from the cells and spore. That's why it can be remain dormant for a very long period of time. A protective agent like skimmed milk of the sugar reduce its effect. DMSO, that is the most potent cryoprotective agent, and glycerol is also a cryoprotective agent. This is a sequential diagram of metagenomics where we can isolate the bacteria, fungus, virus from a environment sample. Then isolate the gene or DNA and then manipulate the DNA, and then after identification of a particular gene of interest, we can clone the gene, and then we try to express the gene in particular model organisms so that we can utilize for at an industrial level if it is useful for human beings. So this is typical representation of metagenomics approach. Next, try to differentiate what is the difference between batch culture, feed batch culture and continuous culture. Such type of question may be asked in your CBSE exam. So try to differentiate between batch culture and continuous culture. In case of batch culture, we can do the process of fermentation in certain volumes after cleaning the fermenters and complete the fermentation process uh, with a limited amount of volumes inside the certain uh, uh, fermenter in a certain period of time in a particular batch. So cultural environment, continuous changes, growth product formation and substrate utilization all terminate after a certain time interval, means there is a completion of fermentation. In case of continuous culture, we can put the certain volume of media, and we can also collect the products that we cannot stop. Okay, so growth in continuous culture takes place under a steady state condition. Growth occur at a steady constant state and in a constant environments. So we have to maintain the growth rate during whole entire process in continuous culture. But here growth rate will be maximum in case of batch culture, then it will come to a steady state, then finally come to declining stage. So after a certain period of time, all process will terminate in case of batch culture regarding fermentation. What are the factor of environment? Factor like pH value, concentration of nutrient, metabolite products, oxygen, which changes during the growth of batch culture. But these all factors remain constant during continuous culture. Now, what are application of microbial cell culture technology? It may come in your coming CBSE exam. Means production of whole microbial cells. We can produce spirulina like algae, and production of primary metabolite like so many vitamins. Production of secondary metabolite like toxin, antibiotics, and exploitation of metabolism. There is a biotransformation reaction, means You can change less useful product into most useful product in a medical field as a medicine, then synthesis of recombinant proteins. So these are the major application of microbial cell culture. Thank you and have a nice day.